Comments before I start. One, I just want to reinforce what Mike just said, what Nina said. Everything we've been talking about today, whether it starts with history and physical that Bill emphasized, to all the new technology that's coming and we're excited about, it's all for the purpose of finding an answer for the patients and families we serve. And it's all to empower them, provide hope, and hopefully eventually prevention, treatment, or cure. So I commend all the speakers today for continuing to keep that focus in everything we talk about. The other comment from the talks today, it was mentioned, I think, throughout the day, but it didn't really get its own session, and that's the importance of bioinformatics and data analysis. I think that's a technology that needs to stand alone, because I don't think any of these are be successful without significant bioinformatic resources and data analysis and computational power. So maybe that's a future conclave at some point. This talk, first of all, a disclaimer or a full disclosure. I didn't pick this title, and then when I was getting up this morning, I said, I don't know if my talk really addresses the title. So for those of you that will be disappointed if I don't address the title, I think I do have one slide that says something about cells, so I'll say something about cell cultures. Animals, when I got up to run this morning, I put on the t-shirt from rhettsyndrome.org, and on the back of it said, scientists have reversed Rett syndrome in mice. It's time we do it in humans. And I've mentioned that just because I think Tom mentioned this. There's a lot of great work that's done in mice and other animals, but it doesn't always translate to humans. So I think we've all heard the comments that we've done a lot of good for mice, but we haven't done as much good for humans. So hopefully someday we will. In children, I did want to mention that because I didn't really emphasize this in my talk, but and I think the reason Roger put that in the title was that so often in regulatory issues and clinical trials and safety concerns that we never get down to testing and putting children in trials because we get stopped at the adult or adolescent stage because of either poor outcome measures or poor biomarkers that we can't show that's effective in adults, and we never get down to the children. I was thinking during Nina's talk that obviously everything she does for the newborn screening is to get to prevention and treatment, and those treatments are being done in children. We've got to figure out how in all the neurodevelopmental disorders that we're getting those clinical trials, the treatment trials down to the appropriate age groups. I think we all feel that if we don't have early diagnosis, early detection, and early intervention, early treatment, we're not going to change the course for those children. So another disclaimer, the majority of this talk is my opinion with very little science, but it's the end of the day, so I guess I could do that. So first, just a few definitions. Clinical trials, treatment trials, are research studies, so it is research. And for the most part, we're exploring safety and efficacy of treatments for humans. Typically, treatment trials or clinical trials follow a very long, laborious period of preclinical research that's done in vitro work and in animals. And there's a high degree of regulatory oversight. Institutional Review Board, Department of Health and Human Services has an Office of Human Research Protection, DSMB, Data Safety Monitoring Board, FDA, and CRO, Contract Research Organizations. There are probably more, but each of those, whenever we do an FDA or NIH or industry-related trial, we have to mention that all of those people can at any time come and review our data, and that they all have their own interpretation of regulations. And what one monitor might think is the right way of doing it may not be the way the next monitor. So it makes it very challenging for those that are doing clinical trials. So having said that, it's very important for us to do these human subjects training programs that are now required, and those that are going to do clinical trials doing the good clinical practice training. So it will come up when you're doing clinical trials. There are many slides like this that you can see, basically just talking about the drug development process, and that it's a long process, and it's a process that has a very low success rate. There's typically a 10 to 15 year period of drug development, and very expensive now. It's at a little over $1 billion for a successful drug to come through this pipeline. And again, 3 to 6 years of preclinical work in the lab with in cell cultures there, I mentioned it, and animals and other kinds of preclinical work, and then frequently 6 or 7 years in the clinical trials, and then still it keeps on going after the compound or drug or treatment is approved in a post approval monitoring in the phase 4 trial. So it's an expensive and laborious and long process, and this just shows that another way. And then you start out somewhere 5 to 10,000 compounds that might be screened as a potential product to bring to market. You go through the preclinical testing, you're down to about 250 that get through the screening process, and then 5 might enter a clinical trial, and you might have 1 that's approved by the FDA. So it's a long process. It's risky for the pharmaceutical companies, for investors, for individual clinicians that are trying to bring something to market. But having said that, if you look at clinicaltrials.gov, there's been a significant increase in the number of clinical trials over the last 20 years or so. Now, not all of these trials are treatment trials. A lot of these may be natural history or device trials or things like that. But over almost 260,000 clinical trials listed on clinicaltrials.gov. I'll mention 2 things. One, there's this International Committee of Medical Journal Editors, and that little diamond means that in whenever that is, 2006, they said that for them to publish any clinical trial data, it has to be listed on clinicaltrial.gov. So just a reminder or warning, if you're going to be doing clinical trials, one reason to put it on clinicaltrials.gov. And then this little diamond, the FDA in I think it was 2008 or 9, said that any trial that's going for FDA approval or that's sponsored by federal funds has to be placed on clinicaltrial.gov too. So that's just another thing that has to be done. But it is helpful for clinicians and families to find what trials are out there, what treatments are out there. So that's a little background on clinical trials. I'm going to talk a little bit about clinical trials here at the Greenwood Genetics Center, and then some comments from that. So first of all, these are some clinical trials that we've done here in Greenwood with the team here, Angelman and Rett syndrome. And the first point I guess I want to make from this is how and why did we get involved with treatment trials for Angelman and Rett syndrome? Well, we're involved in the natural history study. We're involved in a lot of publications and work on that. And I guess the point I want to make out of that is that pharmaceutical companies, FDA is not going to come asking you to be in a clinical trial unless you have patients, one, with those conditions, and unless you're knowledgeable about that condition. So I think this is important for those that want to get into clinical trials, that you need to be developing an area of expertise in certain disorders. You need to be publishing on those. And I will tell you that you don't have to necessarily be intelligent or writing those articles. You just have to hang around with people that are intelligent, like to write articles, and you can still get to do clinical trials. But I think if we want more clinicians involved in those, we do need to be thinking about certain conditions that we're going to become the experts on and be significantly engaged with seeing those patients, evaluating those patients, be engaged with the family support groups and foundations as well. Other comments to make from this. One, this was one of our early trials, the supplement trial with Angelman. We had 21 patients enrolled, and they came from all over the country. I mean, West Coast, Midwest, Northeast, to come here, because we were one of only 4 sites. It was also over 10 years ago, I think, when we did that trial. And I don't know when social media started, but probably before social media was as big as it is now. And there weren't a lot of trials out there. So at that point, patients, families were anxious for treatment. They went wherever the sites were that were giving the treatment, and they were ready to enroll in anything that might help their child. Come forward to this trial, or even this trial, we're seeing, particularly like Rett syndrome, I think Walter alluded to this earlier, we have a potential of 4 to 6 new trials next year. Various pharmaceutical companies, various compounds. Families now are pretty savvy. They know what's coming down the pike, and they're picking and choosing which trial they think would be best for their child based on either what they've read about the compound or what symptoms or the clinical endpoints for their trial. So one of that is it makes it very difficult to recruit patients because they're kind of self-recruiting for the trial and site that they think is going to be best for their child. So and we're dealing with rare conditions, so there's not a lot of patients out there. So it makes it more challenging as you're going forward in clinical trials. Fragile X, we've been involved in Fragile X trials as well, and not really that involved in Fragile X syndrome, but I got into this basically through Charles and Roger when Novartis wanted blood samples on patients to look at methylation status, and we did a good job. We were able to recruit a lot of patients that were willing to give blood samples, but because of that, when they started their clinical trials, they said, would you be interested in doing clinical trials? And we did. And then the point I want to make on this, in the first trial, we enrolled 9, and then you see 3 subsequent trials where we enrolled 0. This is a point that Walter makes frequently, that a negative trial that has a negative outcome can be very detrimental to other clinical trials. So basically what happened, these all were involved with the mGlu-R compounds, and when they did early data analysis, they didn't see a positive effect from the compounds, and so they basically did not go forward within these trials. So we had gone through the process of site initiation visit and getting all the everything in line to start, and then it was pulled out from under us. And so there was a lot of effort going in to get ready for a trial, both for the company and for the site. So I think that goes back to the importance of well-designed trials, well-designed biomarkers, outcome measures, and as I think has been alluded to with the arpacarin trial, there may have been a signal in there, but because it was a secondary outcome measure, not a primary, it wasn't accepted by the FDA and kind of killed the trial as far as the pharmaceutical companies. So a couple comments about clinical trials at the Greenwood Genetics Center. Challenges. One, we don't have a clinical translational science award. This is the NIH process of setting up centers at major medical institutions to support translational science. MUSC has a clinical translational science unit in South Carolina. And theoretically, we are part of that. In the application, we were included as that, but it really hasn't helped us as far as having the resources to do clinical trials. Basically, they have a unit that is there with rooms, beds, x-rays, labs, nurses, all the type of things you might need to do a clinical trial. So basically, at the Greenwood Genetics Center, we have to create that ourselves. When we started this process, the team in Greenwood, we had no experience. It wasn't limited experience. I'd never done a clinical trial before in my life. And there was very limited infrastructure. But we've overcome that and are doing clinical trials and are going through the process of getting some additional trials set up. I thought this was an advantage when I first started, that we had multiple clinical sites that you could enroll patients all over the state and help eliminate some of the travel to different sites. But it actually has been a challenge in that pharmaceutical companies want to come in and do a site initiation visit, and every site is an individual site. And so it means you have duplication of equipment, freezers, EKG machines, everything like that. So it's not one site that is an umbrella over all our 5 sites. So it actually has been a challenge for us to figure out how to do that. So far, clinical trials have been done here in Greenwood. Some things that have been advantageous for us. One is having the lab here and having it right upstairs. It is helpful to have lots of minus 80-degree freezers at your disposal. It's helpful to have centrifuges of any type, and it's helpful to have lab technicians that are willing to help you if you don't know how to do a particular procedure in the protocol for that. We're very fortunate to have a good relationship with our institutional review board. I continue to be impressed with the relationship we have with self-regional healthcare compared to other sites around the country when I hear some of the battles they go through with the IRBs that they have to deal with. Our IRB is very realistic. They do look at safety of patients. They do make sure we're following regulations, but they're also very realistic about how we do things. We also have a very supportive hospital. We mentioned we don't have a CTSA, but we have a hospital that when we need EKGs or EEGs or labs or pharmacy, they make it work, and they've been rules as far as we don't have to go through registration all the time. We don't have to pay the standard rates. So having a supportive hospital has been a critical part of making this successful. And then I can't say enough about the team we have here that makes this work. We all take on positions or tasks in the treatment trial that we've never been trained for, and it may not be in our area of domain or expertise, but we do whatever needs to be done. I say all this just because if other clinicians at other sites want to do clinical trials, it can be done. It is not that hard, but it does take a lot of work from the clinician. It takes a lot of work from the coordinator and from the rest of the team to learn these regulatory issues, to work with the hospitals, pharmacies to get all these things resolved. I can't say enough about having a coordinator that helps with all the regulatory issues. It is very nice when I don't have to worry about that. I just have to sign the documents and trust that they're going to be put in the right place. And we get excellent reviews from all the site monitors. So just kind of looking forward, I certainly think and hope we will continue industry-sponsored trials, though those are obviously directed and decided upon by the industry, though Walter is having more influence on trying to help direct industry into certain directions with trials and make sure they're well designed with good outcome measures, good biomarker discoveries. I think we're uniquely poised here to be involved in biomarker discovery with all the new technologies that were outlined and discussed today. And I think those are going to be helpful in that if we can find biomarkers that predict who's going to respond to a specific treatment, that certainly makes for a more efficient, more effective, and less costly trial. And I know that Tim and Laura and Tarek have been involved in a lot of biomarker development and for trials that we don't do, but hopefully we'll start seeing them involved in more clinical trials that we're doing here. And ideally, you wouldn't need improved outcome measures. Ideally, the results would be so great that you could just tell it by looking at the patient that they're significantly better. But it's not just better outcome measures, but it's the right outcome measures. I think sometimes the things that we're looking at endpoints in these clinical trials really maybe don't make a difference to patients or families, and that if we looked at other outcome measures, it might have more. It might be more important as far as the outcome of the trial. Some things that we do have here that are somewhat unique, the biolog system, whether that is something that can help us with biomarker discovery or developing treatments to see if certain patients may respond to specific novel treatments so that we can actually develop clinical trials here. And it's certainly the hope of what we're doing that, and I think it has great potential for that. Model organisms certainly is something we've not had on campus before. With the Clemson endowed chair coming, we will have Drosophila on campus starting next year. How helpful that will be in clinical trials, I don't know. There are some other discussions that may bring other model organisms on campus, which may help at least with moving forward with functional markers, biomarker development, and potentially useful in clinical trials. We are doing some iPS cell work here and some CRISPR work here, but I think it's very early and very elementary. I think that nationwide we will see these having a greater impact and greater role in clinical trials, and hopefully between iPS cells and CRISPR and gene therapy, we will actually be treating and curing some of the patients. But when I think of gene therapy, I looked into that a good bit about 15 years ago. There was a quote that I always remember, that gene therapy has been about 10 years away from the clinic for the last 30 years, and I think 15 years later we're still saying that. But I do know that we have a speaker coming next month, well, this month, it's November, to talk about gene therapy, particularly for the central nervous system disorder, particularly looking at neurodevelopmental diseases. Hopefully we're getting closer and closer. We certainly know that gene therapy is having some success now, particularly with eye disorders, hematologic disorders, immune disorders. And then this popped up on CNN app on my iPad this morning. Scientists replace skin of boy with rare disease. So this little fellow has epidermolysis bullosus, and they took stem cells from him, and they used a gene vector to put an unmutated copy of LAM3 into his stem cells, and then they grew up skin, and they replaced his entire skin with skin they grew in the lab. And I didn't show the next picture, but now he's attending school and doing well and doing quite well. So I showed this. I wasn't looking for it. It just popped up this morning, but these technologies are happening. They're exciting. They're being applied in a lot of disorders, and it's certainly the direction that we have said for the last, 15 years that we want to be moving in this center. I think to do that, we need to put equal emphasis on clinical trials and treatment, and some of these technologies that the same emphasis that we do on next-gen sequencing or metabolomics or microarray, we need to be putting that same emphasis and resource to that. So this is where the whole field's going. We need to define where we want to be in that process as we move forward. So I'll just leave with that, the excitement of what's happening in the future, Mike.